Alright, glycolysis regulation. So regulation is basically how fast the process of glycolysis is happening. So we're going from glucose to pyruvate, and this is always happening, whether it's happening very quickly or whether it's happening very slowly, or perhaps even going, the other way, gluconeogenesis. Depends on many different things. So if we're going to sum up the regulation in a nutshell, the things that makes this process happen quicker is insulin. Always associate insulin with PP1. And the other thing that helps this process move along is AMP. On the other hand, the things that slow it down epinephrine or glucagon, associate both with pKa, and ATP as well. So remember, the whole point of this is to make ATP. So if you've already made ATP, you can stop making more ATP. So there's a lot in this, the regulation section, which is pretty much half of the PowerPoint. So I found it's easiest to divide it into 6 topics. We're going to talk about what happens in the pancreas, how AMP plays a role, what happens in the muscles, liver sequestration, allosteric regulation, particularly reactions 1, 3, and 10, the 3 regulatory steps. So this by far is the most in-depth, maybe the hardest to wrap your mind around. We'll briefly mention gene regulation. That's no more than a list. So let's get these ones out of the way first. They're not so bad, pretty straightforward, just a little bit of memorization. We'll talk about the concepts of hormonal allosteric regulation. So in the pancreas, it's a good thing to know this. It also comes up in endo. So all these start with glucose coming into the cell. Because we're in the pancreas, we're using GLUT2. Remember, GLUT2 is pancreas, beta cells, or liver. Same thing with enzyme for step 1. It's glucokinase because it's in the beta cells. Glucokinase also found in the liver. And that leads to glucose 6-phosphate, all the way through glycolysis to pyruvate, all the way through the Krebs cycle and oxidative phosphorylation to make ATP. So the ATP, the concentration of that goes up, and the ATP-gated potassium channel is blocked. So if we were going to assign events, glucose entering GLUT2 is event 1, ATP-gated potassium channel blocked is event 2, which causes a depolarization. Then the voltage-gated calcium channels open because of that. More calcium is in the cell and insulin is released. So just know all these steps, know what's associated. His favorite things to ask tend to be the GLUT family. The next topic is AMP. So as muscles contract, ATP is hydrolyzed, and you get ATP, free phosphate, and the energy that comes from that reaction. So you have all these ADPs floating around. You take 2 of them, and you can turn them into AMP and ATP from adenylate kinase. So that's a good enzyme to know. Now that we have all this AMP kinase is on. That's a good one. That one's your friend. It's going to cause glycolysis to happen, more glucose uptake, you're going to be burning fat, and you're going to stop making fat and cholesterol. So there's a chart that shows AMP kinase and its effects. It's a good one to be familiar with. So the regulation of muscle is pretty simple. You either have low energy, which means you have more AMP than ATP, or you have high energy, which means you have plenty of ATP. So if you have plenty of ATP, and the purpose of this is essentially to make ATP, this is going to be inhibiting the process. So the steps that it is inhibiting are steps 3 and steps 10. If you have ATP, that's going to block those two enzymes, or at least slow them down. If you have AMP, that's going to stimulate this enzyme, PFK1, in reaction 3. Now that reaction is going to take place, and then you have fructose 1,6-bisphosphate, and that actually causes a feed-forward stimulation. The presence of this metabolite, for some reason, I can't explain it, it just does, stimulates reaction 10, pyruvate kinase. So the next topic is liver sequestration. Remember, glycolysis is happening in the cytosol, so if we are bringing glucose in from the liver, we're going to use GLUT2, and because it's in the liver, we would use glucokinase. Now, glucokinase is stored over here in the nucleus, and the thing to remember about glucokinase is that it is a glucose sensor. Which means it knows when glucose enters the cell, and it brings glucokinase out to help with reaction number 1 here. So then you have glycolysis taking place as usual, and you get fructose 6-phosphate. This tells glucokinase to go back in. So it's done its job, you have enough glycolysis taking place, so as soon as you get to this metabolite, this takes glucokinase, sends it back into the nucleus. That's all you need to know for liver sequestration. Again, the things to note are always glucokinase because it's the liver, GLUT2 because it's the liver, and this is where the sequestration happens. So there's a chart talking about the allosteric regulation. This kind of sums up everything we're going to be talking about. So you're going to look at which reaction, 1, 3, and 10, the enzymes associated. Note that glucokinase or hexokinase 4 is not regulated allosterically. It's just 1 through 3 that are inhibited by G6-phosphate. And that's a simple negative feedback. You go from glucose to glucose 6-phosphate by hexokinase 1, 2, or 3, and because you've made this, it's going to cause a negative feedback loop here. So we already have enough of this. Let's slow down. There's just one step it's regulated at. So that's all we're going to say about reaction 1. That's the easy one. Reaction 3, phosphofructokinase 1 is activated or stimulated by low energy, AMP or ADP. You can also call that low energy, which means it's going to be inhibited by high energy, or ATP. The most important things to remember about reaction 3 is fructose 2,6-bisphosphate is what's going to activate it the most. So that's the most potent activator, and that happens in the liver. Citrate inhibits this process in the liver. Just memorize that for now, it'll make more sense in the amino acids unit. Step 10, pyruvate kinase, is stimulated by the presence of fructose 1,6-bisphosphate, like I mentioned before. That's just something to memorize. That's the feed-forward stimulation. and it's inhibited by high energy, alanine, acetyl-CoA, and long-chain fatty acids. Let's take a look at reaction 3. It's the most complicated of the reactions. This is mainly happening in the liver. The regulation of this step is mainly happening in the liver. So if we're going to say it in the most simple terms, reaction 3 is going from fructose 6-phosphate to fructose 1,6-bisphosphate. That happens because of PFK1. PFK1 requires a small amount, any amount, of fructose 2,6-bisphosphate. That's the activator of it. You get that because of the presence of insulin. Remember, insulin is because of high blood glucose, and always associate insulin with PP1. Insulin essentially is stimulating PFK2, which is making this. So we'll take a look at that here. So this sometimes confuses people because there are two enzymes happening in one protein. It's a bifunctional enzyme. So both PFK2 and FBPase2 are on the same thing. Just one is going to be turned on and one is going to be turned off at all times. So when PFK2 is on, FBPase is off. And remember, it's on because of insulin, PP1. PP1 is a phosphatase. Remember, phosphatase is dephosphorylate. So that's why there's an OH here. The other half of the picture is you can turn off PFK2 and turn on FBPase2 by epinephrine or glucagon via pKa. So when pKa is active, it's going to phosphorylate the regulatory domain, turning FBPase2 on and PFK2 off. So you'll discuss later when FBPase2 is on, that's when gluconeogenesis is happening. But for now what we're worried about is insulin stimulating PP1, which turns PFK2 on by dephosphorylation, and PFK2 makes the F26 biphosphate. And that's what is the most important thing in reaction 3. Here's a generic example trying to better explain how these enzymes are allosterically regulated. So these enzymes, which are proteins, have these OHs sticking out. And they usually come from serine, threonine, or tyrosine. And that's your polar group that all have the OHs. And they can be in two positions. They can be dephosphorylated, so they just have their OHs hanging out there, or they can be phosphorylated. So in this unit, kinases are what is phosphorylating the enzyme. So kinases, mainly pKa. Phosphatases, mainly PP1 from insulin, is what's dephosphorylating the enzyme. Now there's no easy way to remember, the on-off position. Some enzymes are on in this position, some enzymes are off in this position. It all depends on the enzyme. So if some may be on here, which would make them off here, but it could be the other way around. Pyruvate kinase is this way, so we'll take a look at that enzyme for step 10. So far we have seen PEP go to pyruvate. It's also good to note that PEP. It's PEP plus ADP plus a proton is what gives you pyruvate and the ATP. This happens because of pyruvate kinase. The other things that we learned are the feed-forward stimulation, fructose 1,6 biphosphate, the presence of it activates pyruvate kinase, and ATP, acetyl-CoA, alanine, and long-chain fatty acids slow this process down. So pyruvate kinase is turned on by insulin, or PP1. So it's in its on position when it's dephosphorylated. This can be a serine, the threonine, the tyrosine, amino acids. It doesn't matter what it is. The only thing that matters is that it's dephosphorylated and it's on. So insulin speeds up this reaction. It allows glycolysis to continue. pKa from epinephrine or glucagon phosphorylates pyruvate kinase. It attaches the phosphate group to where that OH residue was, and that's what turns it off. There's a little section about gene induction on one of the slides. It comes up more in other units, so I wouldn't be surprised if it's asked about it. I can't explain it that well at this point. It'll make more sense in later units. For now, I would just know that the enzymes and reactions 1, 3, and 10 are all involved in gene induction. He specifically mentions just the hexokinase 2 version and the glucokinase, but I don't ever remember him explaining why. It's just those two and not 1 and 3 here. So I would just memorize the list to play it safe. Our enzymes from reaction 3 and pyruvate kinase.